We've talked about the fact that proteins bind and that that's part of their function to bind. Enzymes are proteins that not only bind, they catalyze. They lower the energy barrier for a specific reaction. If we look at the transition state, that is the energy of the state that is the intermediate in the reaction, the activation energy without enzyme can be quite high and can present a significant energy barrier to a reaction. On the other hand, enzymes lower that activation energy so that the mountain that has to be climbed to get to the products is much less. And so reactions that are very slow without the enzyme, because transition state determines rate, the activation energy determines the rate of a reaction, you can very significantly enhance the rate using enzymes. Note, however, that the overall energy that's released during the reaction is the same, independent of whether the enzyme is present or not. So the overall energy is unchanged. What is changed in the presence of an enzyme is the rate of reaction. It is a catalyst, and remember that catalysts are regenerated during the reaction, so that the enzyme binds to the reactants and releases the products, and it is unchanged and can do that again. So it can do it over and over. We need to talk a little bit about Gibbs free energy, because the equilibrium, that is the concentration of reactants and the concentration of products, give you an equilibrium constant. That equilibrium is not influenced by enzymes. It is the activation energy that is influenced. So if we have the reaction C plus T going to A plus B, if we write the free energy equation, we have delta G is equal to delta G naught plus RT natural log of the products of the concentration of the products and the product of the concentrations of the reactants. But at equilibrium, delta G is equal to 0. The system can do no work at equilibrium. That means that delta G naught is equal to minus RT log K equilibrium, which is the products of the product of the concentrations of the products at equilibrium over the product of the concentrations of the substrates at equilibrium. And if we write delta G out in this way, we'll see delta G is equal to the equilibrium, natural log of the equilibrium constant, and the natural log of the concentrations at any given moment. But it turns out that delta G naught is a very useful way to think about how much energy is released or taken up in a reaction. In general, the overall delta G has to be negative to proceed in the direction a reaction is written. The delta G naught and the concentrations of the reactions have to be considered to determine whether a reaction will go in the direction that it's written. But again, as an initial estimation, the delta G naught, which is related to the equilibrium constant, will give a sense about which direction a reaction is going to proceed, and that's often used. It turns out if you couple reactions, you can use the energy of one reaction to pull reactants in the direction of products. And in particular, coupling hydrolysis of high energy compounds like ATP and GTP, which we will see over and over again as we go along in this course. So let's look at this reaction. If we have A going to B plus C, we have a plus 5 kilocal delta G naught. This reaction is not going to proceed spontaneously. If we have B going to D and we have a minus 8 kilocalorie per mole release of energy, reaction 2 will proceed strongly to D. And if we add these together and get reaction 3 of A goes to C plus D and B is produced and then used in the series, with coupling reaction 3, A will proceed to C plus D. And this is the kind of coupling that occurs in biological systems and often in enzyme complexes, as we'll see shortly. So here's an example. The overall reaction is glucose plus ATP goes to glucose 6-phosphate plus ADP. Actually, this is two different reactions. One is glucose plus phosphate goes to glucose 6-phosphate plus water. That is an energy requiring reaction. And ATP plus water goes to ADP plus inorganic phosphate, reaction 2. That reaction proceeds spontaneously. It has a negative delta G. So the energies are additive for these overall reactions, so that delta G for reaction 1 plus delta G for reaction 2 is minus 3 kilocalories per mole. So by coupling these reactions, you can actually create glucose 6-phosphate along with the hydrolysis of ADP. And this linking of reactions is a way to drive reactions to completion. It's a way to capture negative free energy of one reaction to make it possible for the other reaction to occur. And again, think ATP hydrolysis. We'll see this over and over to make reactions move to completion. You can not only capture the negative free energy by coupling reactions, you can alter concentrations of reactants and products to drive equilibrium. And one example of that is in a pathway, one product becomes the substrate of the next reaction. And so you have A plus B goes to C plus D, D plus E goes to F. And so you have a consistent depletion moving in the direction of F as long as this overall energy change is negative. As long as the overall delta G is negative. And so by depleting substrates from one reaction and using them in the next reaction, you can drive pathways to a particular product. So if you stop and think, there are a lot of reactions that go on in the cell. How many different types do you think actually occur? Hundreds? Thousands? More. And all of them are using this fundamental strategy of stabilizing the transition state.